Hello and welcome to the photosynthesis webinar series. I'm Louise Diva and this webinar series is sponsored by the Characterization project. I'm delighted to welcome my two speakers today, Professor Wenbin Zhou and Professor Stefan Schilberg, both of which are going to talk about improving photosynthetic efficiency in crops and how they have achieved or achieving this. As always, we like these to be interactive webinars, so please put your questions in the question box. At the end of the talks, I will present them to each of the speakers. If you've got a burning question, it might be a bit difficult to put it into the questions or by text and you'd like to ask the question, you can raise your hand and if we can unmute you, we will. So on to our first speaker. Our first speaker, Wenbin, leads the Regulation of Crop Photosynthesis Group at the Institute of Crop Sciences in the Chinese Academy of Agricultural Sciences in Beijing. He's interested in physiological and molecular mechanisms of photosynthesis to improve photosynthetic efficiency and yielding in crops. And he's going to talk about some of this work he's done improving the yields of rice, achieving quite substantial yields from what I've read in his paper, which if you haven't read, is really good. So I'm going to hand over to you, Wenbin.
Fine.
Yes.
Yes. Okay. Okay. So firstly, I would like to give a very brief introduction about global food security. So as we know, that the rapid increase in human population, shortage of arable land, climate change, especially the occur for COVID-19 pandemic, and international conflicts that has swept the world in the past 3 years. So these years, the FAO just released the latest research report. According to this report, about one third of the world population still face food insecurity. So Wenbin, on the other hand,
Wenbin,
okay,
are you changing your slides, or I think we're stuck on the first slide, the title. Is that all right? We're just on the first.
No, I'm focused on the first slide.
The slide, yeah.
Yeah.
Good.
Okay. So on the other hand, in recent years, yet the improvement of major crops has been stagnated. This is reported by many public reported in many literatures. So with limited possibility for increasing arable land and global food security is currently facing severe threats. Okay. So therefore, there is a urgent need to find a sustainable way to produce more agricultural products on limited arable land and also ensure food security in the future. Actually, in my opinions, crop high yielding is a forever topic of agricultural production. So meeting this challenge will require the development of new crop breeding, genetic engineering, and also cultivation strategies. So far, two breakthroughs have been made in the last centuries. So the first breakthrough is the Green Revolution in the 1960s. So it was mostly driven by adoption of semi-dwarf varieties of the crops. So this is due to the introduction of SD1 genes. Okay, so the exploitation of heterosis by introduction of hybrid rice in 1970s marked the second breakthrough and the second leaps in the crop production. So these achievements of the 1960s and 70s lead to the increase of rice and wheat in many countries. And every breakthrough is closely related to the finding of the utilization of a gene resource. Okay. So on the other hand, the massive production of application of nitrogen fertilizer has been widely used to maintain high yield of crops in agriculture over the past centuries. However, only a small fraction of the applied nitrogen is absorbed by plants. A large portion of it is lost to the environment and cause serious environmental pollution. So, as I said before, crop yield improvement of crops has been stagnated. So, how to further improve the crop yield? As we know, the photosynthesis supports the growth of plants. Okay, so in recent years, the improved photosynthesis has emerged as a promising strategies to increase crop yield, and is seen as the next frontiers in achieving crop yield improvement. So, different strategies to improve crop yield has been proposed, such as introducing the C4 photosynthesis pathway into C3 plants, introducing the algae carbon concentrating mechanisms, and also modifying metallic pathway, and so on. So I just listed here. Okay. So C4 photosynthesis is more efficient than C3 photosynthesis for two reasons. A repertoire C4 enzyme and leaf anatomy with reduced photorespiration. So by doing so, C4 plants can lead to the increased photosynthesis. This allows a faster biomass accumulation with a higher nitrogen and water use efficiency to the C3 plants. So I just show you an example. Okay, so the maize golden tool genes was the first regulator to be identified. So G2 encodes a MIP-related transcription factor of the GAARP family that is expressed mainly in the bound sheet cells, whereas the related GLAC1 is expressed mainly in the mesophyll cells. Okay, so as reported, chloroplast development has been activated in the bound sheet cells of the rice through overexpression of the G2 genes. So recently, a study from our group collaborated with the Janlander's group. Okay, we found that field-grown lines overexpression G2 showed up to 30% of yield increase with reduced photoinhibition and increased photosynthesis. Okay, this example validated the overall strategy that is a transition to the C4 can be achieved in C3 plants. So as we know, the carbon and nitrogen metabolism are the basis for life on Earth. So nitrogen is the major component of organisms and the critical constraint of crop yield. Meanwhile, photosynthesis is the source of primary productivity for plants, providing carbon skeletons, energy, and reducing power for nitrogen assimilation. So therefore, the balance and the trade-off of carbon and nitrogen metabolism are critical to the all plant growth, development, and also for yield. In 2014, a paper published in Nature Biotechnology, so using comparative analysis of maize and rice leaf transcription and metabolism, has been identified a set of 118 transcription factors that regulate nitrogen and carbon metabolisms. So they may act as a regulator of C4 photosynthesis. So based on this research, and our concept on carbon and nitrogen coordinations, we screened this transcription factor for their responsive to light and low nitrogen in rice. So firstly, we conducted the RNA sequencing analysis in rice plant growing under low and high nitrogen conditions. So we screened 13 genes showing the greater induction has been identified under low nitrogen conditions. Secondly, so investigating of the light regulated mRNA accumulation showed that 1 of 5 genes, so the OSDREB1C genes, so encoding a DREB type transcription factors, so previously showed to be inducible by abiotic stress, so displayed a dual profiles. So with the expression increasing with the duration of light exposure, so we are very interested in these genes. So to further characterize the function of these transcription factors, so we generated several lines of overexpression lines and also knockout mutants. So in the Oryza sativa Nipah genetic background. Okay. So we observed faster growth of overexpressed plants at the seedling stage. So whereas the growth of knockout seedlings was retarded compared to the wild type. So in addition, so we also noticed that the overexpressed plants displayed longer shoots. Okay, we also detected no growth difference among the wild type overexpression and the knockout plants when the seedlings were cultivated in the dark. So this data suggests that a light-induced mechanism of growth improvement. So this may be give a hint for photosynthesis for these genes. So next we check the photosynthetic machinery. So you can see here. So in the overexpressed plants, so you can see increased pigment contents, including the chlorophyll and the carotenoids. And you can see the increased accumulation of a photosynthetic protein complex. And also, you can see both chloroplast number and size, photosynthetic protein complex, and both ribosome content and activity will increase in overexpression plants. So the other way around in the knockout mutants. Next, we evaluated the role of the DREB1C in regulating photosynthetic capacity. So you can see here. So photosynthetic parameters, including dual change, light response curve, and the CO2 response curve of net photosynthesis, were improved in overexpression plants. So this is in agreement with the higher maximal rate of ribosome carboxylation and also the higher maximal rate of electron transport. We also observed that the accumulation of a high amount of carbohydrates in leaves and lead to the improved grain filling rate and also 1,000th grain rate. So next, we also performed the nitrogen application experiment to check whether nitrogen use efficiency increase or not. So firstly, we incubated the seedling with the nitrogen nitrate solution. After 3 hours of incubation, the overexpression seedlings had higher nitrogen-15 content in the shoots and also roots compared to the wild type and also knockout plants. We also additionally the overexpression plants increase both nitrogen uptake by roots and transport activity from the roots and shoots. So this resulted in elevated nitrogen content and protein abundance in leaves of the field-grown plants. Next, we also performed a set of field traits with different level of the nitrogen fertilizer application. So you can see these figures. So the nitrogen use efficiency of overexpression plants was increased. Okay, and as shown in these figures, so grain yield and the nitrogen use efficiency of the overexpressed plants was significantly higher than the wild type and also knockout plants across all nitrogen fertilizer conditions. So these results suggest that the increased level of the OSDREB1C confront increased grain yield with less nitrogen input. So we analyzed the carbon and nitrogen distribution in different organs. So you can see here. So the analysis showed that the overexpressed plant accumulated more carbon and nitrogen in the grains, but less in the nitrogen transport to the airway organs. So this is suggested promoted resource allocation from leaves to the shoots from leaves and shoots to the grains. So very interesting. If you can see the finger at the heating stage, you can see here in the overexpression lines, you can see a high amount of nitrogen content and also the higher high amount of carbon content. However, if you can see see these fingers at the maturation stage. So in the overexpression plants, so the carbon and nitrogen content decreased in the overexpression plant. And very interestingly, the overexpression plants showed pronounced earlier flowering phenotype in our field trials. So every flowering was accompanied with the higher biomass accumulation at the heading stage. So this always plant flowered 13 to 19 days earlier than the wild type in Beijing. However, the knockout plant flowered 6 to 7 days later under the long day conditions in the field. Okay. So finally, we checked the grain yield in the field. So we did a lot of field trace experiments in different locations for successful years. So this figure just show you a picture in Beijing. So you can see the grain yield increased significantly in overexpression plants. So the grain yield per plant increased by 40 to 60%. So grain yield per plant is 40 to 68% increase compared to the wild type. Okay. And we'll check the detailed phenotype. So you can see here. So the higher yield of the overexpression lines was mainly attributable to the enhanced grain number of per panicle and also increased the 1,000 grain weight. So this trait apparently resulted from the increased second branch number and also grain thickness, grain length, and grain width, and also grain densities. And interestingly, the grain quality trace was enhanced in overexpression plants, so suggesting that the yield improvement does not have a quality penalty. And we next want to characterize the molecular function of this gene in more detail. So you can see here that the OSDR-EB1C was expressed in all rice tissues and also in different growth stage. The gene expression peaked at the tiller stage, and this gene was expressed widely in all rice tissues, with particularly strongly expressed in the root and the leaves. And the subcellular analysis showed that these genes mainly localized to the nucleus and the cytosol. So we also to check this OSDR-EB1C is whether it's a transcription activator or not. So we use the East1 hybrid assay to verify the direct binding of the DREB1C to the DRE, CRT, GCC box and G box cis element in vitro. And we also done many experiments. For example, the transcription activity. So measurement of the transcription activity showed that this protein was able to activate the transcription of the gas reporter genes. So that means OSDR-EB1C is a transcriptional activator. So next, we want to identify the genome-wide binding sites of this protein in vivo. So we performed the ChIP-seq experiments. So this experiments so identified a total of 10,000, around 10,000 putative OSDR-EB1C binding sites. So among this binding sites, 68% localized to the gene regions and 32% to the intergenic regions. And we next analyzed the differentially expressed gene between the always present and the wild type plants. Okay, so then we extracted the differentially expressed gene with the OSDR-EB1C binding peaks. So in this way, so we totally identified 345 gene were as a putative OSDR-EB1C target genes. So we did the gene ontology enrichment analysis. You can see here. So many pathways had been identified. So for example, transmembrane transport, nitrogen component metabolic process, a primary metabolic process, and also respond to stress regulation of flower development and respond to light stimulus. So for example, if we focus on the transmembrane transport, so the two nitrogen transporter gene identified. For example, the OS NRT 2.4 and OS NRT 1.1b, and also if you focus on the regulation of flowering pathway, there's one gene identified for the OS FTL1 genes. So very interestingly, so if we focus on the primary metabolic pathway, so we also identified a key photosynthesis-related genes, OSRBCS3, so including the rubisco small subunit. So we also know to be transcription activated by light, so was also upregulated in the always present plants. So and also there's one protein, one gene we identified, the OSNR2 genes. So next, we just to check OSDR-EB1C can directly activate these 5 genes. So we're using the chip qPCR and EMSA and also DAP-seq, DAP-seq and ACIS in vitro. So to confirm that OSDR-EB1C binding to these genes. So as you can see here, so this OSDR-EB1C can directly activate these 5 genes. And finally, so we asked the questions. So whether the OSDR-EB1C, DREB1C genes, whether can increase the grain yield in some elite rice germplasm. So we just transformed, we just always present OSDR-EB1C genes in elite rice varieties. So the Xiushui 134. So Xiushui 134 is elite rice varieties in China. So you can see here, we just same as in the Nipah bear genetic background. So if always present this gene in Xiushui 134, so you can see the grain yield increased significantly. Okay, so increase the grain yield by 30% compared to the wild type. And also flowered 2 days earlier in Hangzhou. So this is the same as the case in always present in Nipah bear genetic background. Okay, so in this always present plant, you can see the grain number per bannic, and also setting percentage, and also 1,000 grain weight increased significantly in always present plants. And also to determine whether the function of DREB1C is conserved in other plant species. So we also generate transgenic wheat and Arabidopsis plant overexpression these genes. So you can see here, so an analysis of this transgenic line show that this gene overexpression in the wheat cultivar filter also improved the photosynthesis. Okay, and also reduced the time to flower by 3 to 6 days. Okay, and also to increase the grain yield per plant by 17 to 22% in the field. And so we also did we also performed the growth transgenic plants in the greenhouse. The same case, same as in the field. Okay. And likewise, the transgenic Arabidopsis lines overexpression, also DREB1C had more and bigger leaves. So overexpression this gene in Arabidopsis also showed flowered up to 4 days earlier than the wild type. So finally, to give a summary, by screening candidate photosynthesis-related transcription factor in rice, so we identified a DREB family member, the OS DREB1C genes. So in rice, in which expression is induced by both light and low nitrogen conditions. Okay, we also show that the OS DREB1C driver diverse transcription program determine photosynthetic capacity, nitrogen utilization, and flowering time. So field trials with the overexpression rise and rebuilt yet increase. So in addition, shorten the growth duration and improve the nitrogen use efficiency and promote efficient resource allocation. So therefore, providing our research providing a strategy toward achieving much needed increase in agricultural productivity. Okay. So at the end, so I would like to thank our lab members. So Dr. Shaobo Wei and Xia Li. So they did most of the experiments, and also some experiments were done by our collaborators. And this work was funded by Chinese government and also CAS. So thank you so much. I'm happy to take some questions.
Yeah, thank you, Web. Thank you for sharing your beautiful data. It's some of the most beautiful data I've seen. I really enjoyed it. And actually, 40 to 60% increases in yield. I mean, they're amazing. Does anybody have any questions? Please put them in the question box. We've got a short amount of time left, but we can still take some questions. So I will ask the first question though. So these 40 to 60% increase in yield, higher nitrogen, so I'm presuming higher protein contents, is that right, in your grain? Yeah. Increased nitrogen use efficiency. What's the downside? Is there a downside? Is there any negatives to these plants? They sound like the answer to all the problem, you know, to everything. Is there a downside? Is there any negative effects?
Actually, we didn't see some negative effects. So there is maybe probably as a negative effect. Negative effect is because the grain yield is increased significantly. Okay, so at the maturation stage, so the plants maybe suffer the lodging problems. Okay. Yeah. So you know what I mean? So because the grain yield is too increased so much, and it's too the plant cannot stand the.
So do you need stalk investment? Yeah, you need more stalk investment, which is I think you had the ultimate, but actually it's almost too much, isn't it? And what about your phosphate use efficiency? Is that also improved, or did you look at that? You maybe said it and I missed it. For phosphate use.
So, pardon?
You looked at nitrogen use efficiency. Did you look at phosphate use efficiency as well, or?
We didn't try. We didn't try.
Okay. I've got some comments saying it was very nice work there. Oh, they were beautiful, beautiful graphs and data actually. Let me see, have I any other questions? Is this something you could look for natural variation in? There's like a lot of projects that look at natural variation to try and find these overexpression of these transcription factors. Is there evidence that that's something that would happen for most crops, or?
So yeah, very good question. Yeah, you're right. So we also did the natural variation. So yeah, we did find some nucleotide polymorphisms in the promoter region of the genes. So yeah, we are doing some gene editing experiments. So for this promoter region. So yeah, recently we just got some transgenic plants. Yeah, we got some very interesting transgenic plants. So with also increase the grain yield. Yeah, we're I think that the nucleotide polymorphism so is yeah, we can we did see some difference. Okay, so different in rice varieties.
Yeah, so I think that answers this next question here. Do you see a non-GM approach to apply this knowledge to increase photosynthesis? Do you see? And I think that's what you were saying.
So what do you mean?
I think that's a similar question to what I'm asking. So do you see a non-GM approach to apply this knowledge? So other than using transgenics, is there a way you can do it in a non-GM way, like by conventional breeding?
Yeah, so that's a good question. Okay, so yeah, we just want to have a non-transgenic strategy to create some like rice or crop varieties. So we are doing this, but I think it is possible.
I really enjoyed both the talks. Just to remind you all, our next webinar will be in February. We're taking a break for a while. It'll be in February 2023 on the 7th. And we have two speakers from Australia. We have Bob Furbank and Maria Makova. Because they're presenting from Australia, that would be the middle of the night there at 2 o'clock CET. We're going to be moving the time to 10 o'clock in the morning CET. So please join us for this one. If you joined last month's webinar to listen to Dario Lester's talk and he had problems with his Wi-Fi, that has been re-recorded and it is available for you to view. If you log into the Capitalize website, capitalize.eu, you can find these, as well as all of our previous workshops on field trials, phenotyping and forward genetics. So have a look and see what you'd like to view. It just remains for me to thank you, the audience, for attending and asking lots of questions, and to our speakers for presenting two beautiful talks that I really enjoyed. Thanks very much. I'll see you again in February. Bye-bye.